practice MCQ, and basically there is a Costanzo USMLE review. It used to be the #1 bestseller. I don't know if it is anymore, but it's a review of physiology. They have MCQs at each organ system. Also, I would suggest that your class representative talk to the second year. They have an entire test bank, an entire MCQ bank for the last 10 years on all of the blocks we cover. So all the MCQs are there. And if they didn't give it to you, boy, I would be angry because someone gave it to them, you see. So they should give it to you. You have a test committee or something, at least they did. So they have, yeah, they have it. You have the MCQ, okay? Now, for Hashim's lecture, obviously he wasn't here before, so practice questions were pretty important there, okay? The TBL, I'm going to try to focus the TBL on issues as opposed to, it's not going to be a simply review. We'll try to focus them, and I'll let you know Wednesday because the committee has to approve it, the TBL committee, and they meet tomorrow after my lecture. So maybe Wednesday evening, I'll have something to put up in terms of objectives. Okay. We're going to cover the first part. This is slightly revised from what is posted on Moodle. And sometimes I do that, but I'll repost this again. And what you have, if you downloaded already, it's okay, but this will be more helpful maybe. Today, we're going to be covering the first part, or part 1 of ingestion, swallowing of food. Basically, we'll talk about preparation of the food bolus and the role of saliva, etc. Things that are important for later swallowing, okay? And the pages here are underlined. I'll try to keep up with that if I can. Otherwise, just go to the glossary or index, and you can find these topics in any textbook, actually. Our drive to seek food, or to intake food, or to eat, is regulated by hypothalamic center. I wouldn't worry too much about ventrolateral or ventral medial if you're not a neuroanatomy type class. But just to show you that there's a feeding center and a satiety center. Satiety means, you know, you're full, you're happy, okay? And the feeding center, and they just says, obviously they didn't do it in humans, they did it in animals. But if they stimulate the feeding center, the animal overeats. Hypermorphagia means to eat, right? The terminology, hyperphagia. Then if they destroy the center, that's why they can't do it in humans, they become hypophagic, or they will actually stop eating to the point of death. They're just moaning, and then they die, anorexia. Okay. And the satiety center operates in the opposite manner. If you stimulate it, you lose appetite, okay? They call it oxygenic or something. But anyway, just the opposite. And if you destroy it, it becomes hyperphagic, and they become obese, okay? Excessive stimulation of this center, morbid obesity. We'll get into that in a minute. All these two centers are regulated regulated by input from other organ systems. And we'll be worried about the GI tract mostly, and a little bit on adipose tissue later. But short-term regulation, that's mostly the GI. And what you got here, I got them labeled 1, 2, 3, and 4. And 1 is simply food entering the stomach, induces distension, distend the stomach with increase. And that triggers afferents in the vagus, and they go up and they stimulate the side. You have a sensation of fullness, or you're happy, okay? Has nothing to do with nutrients per se. This is reinforced by certain peptides that are released. We won't deal with peptide YY too much, but cholecystokinin, we mentioned that. One of the peptides we're dealing with, and that actually is a product of the eye cell, of the EEC, enteroendocrine cell, and activates the afferent and the vagus, and it goes up and stimulates the side. Okay. And then you have nutrients themselves as number 3. That reinforce it, prolong it, if you will. And like, for example, if you're absorbing glucose, you have a hyperglycemia. That's a higher glucose concentration in the blood, right? You're absorbing this glucose. And it'll trigger satiety, okay? And it acts directly on the satiety center, the receptors for these things. We'll deal with that later. And the same holds true for amino acids and also for lipids, okay? But nutrients will also induce satiety. The one thing I've got here is ghrelin. It's released by the stomach, but it's not released during feeding. It's released between meals. And some people call it the hunger hormone because it stimulates feeding, okay? So it's cyclic. And once you feed, it goes down. When you're not feeding, it's starting to go up. It's cyclic. Okay, that's short term. Long term, the reason I have it, well, what am I doing? Sorry about that. At least you can see the notes. Long term, oh, now I can see it. Long term involves leptin, that's the red here, and that's released from adipose tissue. It's not necessarily GI, but it has an impact on feeding. And leptin is released by adipose tissue in response to an energy store here. Fat cells are going to be a major storage form of energy, right? So as you increase the number of fat cells, they proportionally increase more leptin, okay? And you'll have a sense of satiety, okay? And then leptin also works by inhibiting ghrelin. It comes over here and inhibits this. So it's turning off your desire to feed, and it's turning on your sense of fullness or satisfaction or satiety. And if they genetically deplete, you can't do it in humans, but in mice, they deplete leptin, these might become morbidly obese, okay? And we're working on that because obesity is a problem in the developing countries particularly, okay? All right, there's also a thirst center somewhere in the anterior hypothalamus. We're not going to worry about it, it's called a thirst center. But the primary regulatory here is osmosis, okay? There's osmoreceptors in the thirst center. How they know that is they injected hypertonic solutions in here, and it stimulated seeking water, thirst, okay? You know what hypertonic is, right? You know what isotonic is? The same osmotic particles as in plasma. Usually that's what we're talking about. You didn't get this in your foundation? You got it, right? So, okay. So once let's say blood or whatever becomes hypertonic, it stimulates the thirst center. You drink water so that you reduce the tonicity, right, to normal. Does that make sense? There's another component in here, just remind you that the GI tract is outside your body. When you lose blood volume, look at this, I block this out, take a look at it if you want, but we'll get it in cardiovascular. It's a deal with reduction of blood volume. But what it's going to do is going to reduce salivation. Now, does that make sense to you? If you're losing blood volume, the fluid for saliva has to come from the blood, because now saliva is outside of your body. It's inside your mouth, but technically it's outside your body. So to conserve blood volume when it's going down, the salivary flow will cease. Okay, it will stop because it doesn't want to lose any more water. All right, a little reminder there. Now, in terms of the rest of this, we're going to be dealing with salivary gland. You're probably going to send me nasty emails after today, but we have the parotid gland and then the sublingual, submandibular. You probably got this in anatomy, I think. And the parotid is primarily responsible for protein and water and electrolytes, okay? whereas the sublingual, submandibular gland secrete mostly mucus. Okay, so these are mucus, and then make a compound gland, and then there's about 600 to 1,000 minor salivary glands that probably produce mostly mucus as well. We won't get into that. 90% of saliva is generated by these three glands. Acinus or grapes. Looks like grapes, I guess. The acinus is this part. You got this histology, right? You got this isn't foreign, is it? At any rate, you have the acinus here, and that results in the primary secretion. It's the initial secretory portion of the salivary gland. The myoepithelial cells, which seem to cover the intercalated duct, and the acini seems to be important for moving the material into the duct system. Now, in the ducts, primarily striated duct, the salivary secretion, primary secretion in the acinus, is modified the ionic composition. So we're going to have to deal with transporters, right? You can memorize this. You don't want to do that, do you? I don't know. At any rate, let's just go over it. Why? Because you'll see all these things again. And that's what I meant. By the time GI block ends, these things are going to be second nature, hopefully, to you, okay? As long as you're sort of paying attention, keeping up. Now, we know this part over here. I'm not going to talk about it very much from now on. It's just sodium ATPase, right? You know what that is, right? It generates electronegative inside the cell, etc., more sodium out, but it generates an electrochemical gradient for sodium, and it's always going to be important. Okay, every cell has sodium ATPase. It's going to be the driver. All right, so I have number one. First of all, let's say that this secretion is chloride driven, and you'll see in the intestine, secretion is also chloride driven. In other words, you're going to get this again and again. So number one, now I covered this, day before yesterday, whatever it was, basic concept. This is a unique transporter. Every time you want to move chloride, secretion, acid secretion, whatever, you're going to need chloride. And the favorite transporter is going to be the one that moves the most chloride for every turnover. So this particular transporter, where is that? Can you guys see that? This moves one sodium, one potassium, two cation, or two chlorides, two anions. In other words, this system doesn't disturb the potential, okay? It's moving negative and positive charges. I'll come back to that. But anyway, you move a lot of chloride, and it's driven by the sodium, potassium ATPase. You're removing sodium, it drives this one, okay? And that's what we meant. Chloride driven, you're going to need this transport, number one. Okay. Number two, that's over here. Chloride channel. I'd like to say it's Cftr, cystic fibrosis, transmembrane conductance regulator. We'll just call it Cftr. But they haven't proven it yet. But at any rate, it moves chloride and somewhat bicarb. Let's say it moves chloride, okay? So this chloride coming in goes into the lumen, okay? This is the interstitium, or sometimes you'll see blood up here, serosa, okay? The outside, well, salivary gland doesn't have serosa. We don't have blood, this will be a lumen of the acid here. All right, and then bicarb can move through here, and then potassium has channels, there's one here. And water. Remember I mentioned aquaporin? We're not going to be worried about whether it's aquaporin 4 or 5 or 6. Obviously, there's a lot of them, right? But it's an aquaporin. It's a channel that allows water to move. Okay. And now we have an exchanger here. This is called the sodium hydrogen exchanger. You notice what's being exchanged here. A cation for a cation. Whenever you run across exchanges, and you will a lot here, you're going to be moving either a cation and exchanging a cation for a cation or an anion for an anion. And when you're moving something together, like take a look at this, you're going to be trying to maintain electroneutrality. Okay, so if you're moving bicarb, you're going to be moving some other sodium, let's say, inside, co-transported. Exchangers, same charge, co-transporters, opposite charge, to maintain neutrality. This is a co-transporter, you got two positive, where's my arrow? Two positive sodium potassium and two negative. Make sense so far? By the time we're done, this all should make sense. Now, also sodium and water. Now, because it's chloride driven, you have chloride coming in here. And later on, we'll talk about sodium movement. But what you're doing is creating an electronegative gradient. Chloride moving in here, it's chloride driven. Sodium just follows across the tight junction. So does water. Remember, anytime you move any solid, water is going to move. That's the way it works, okay? So the net effect is, and we like the terms, I don't think any textbook says it. You use terms like plasma light. In other words, anions are roughly close to what's in plasma, okay? And nearly isotonic, very percent isotonic, okay, in the salivary. Now, this particular system here, you're going to see it very often. You're familiar with carbonic anhydride, right? No? You must have had it before. But this is the way it works, and you're going to see it ad nauseum throughout GI, respiratory, kidney, anywhere. But basically the way it works is, okay, let's say you're moving bicarb. You know, if you know how it works, you know it's going to make carbonic acid, which just associates into proton and bicarb. This is an acid, this is a base. So if you're moving bicarb out, the cell wants to stay neutral. Doesn't like acidification. It doesn't like alkaline. Okay, either way, it's detrimental to the cell. It's not going to survive very long. So if you are moving bicarb through this, you're going to move the proton out. And usually to do that, you're going to be using a sodium proton exchange, sodium hydrogen exchange. These are in the notes, by the way. If you're following this as I'm speaking, you should be okay. Notes say the same thing. Now, in the duct system, you're going to modify this. Okay? And I'll come to why it's complicated. But we have to introduce these transporters somewhere. So you're going to get them all the time. So you might as well start learning about it. One, this is called Epithelial Sodium Channel. I define it in the bottom, but basically that's what it stands for. E for epithelium, and A for sodium channel. C, okay? It's in the notes. You don't have to take it. But at any rate, what it does is sodium enters through this channel. Why? You notice I don't no longer deal with it because that's sodium ATPase, right? It's always going to be there. It's going to create an electronegative gradient for sodium, right? So if you open up this channel, guess what? Sodium's going to fly in, right? So that's the way you absorb sodium. And now potassium, well, you've got to get rid of the potassium, so they throw in a channel here for potassium. All right. So now, well, finally we get to Cftr, which is a chloride channel. All right. But let's take a look at this. The bottom line here is going to be sodium chloride being reabsorbed, potassium bicarb being secreted. We'll come back. It's written right here. And why I have a little box around here, there's an, I guess it's a hormone, aldosterone. You'll get it in the kidney, you'll get it in cardiovascular. It plays a minor role in the salivary gland, but does the same thing. It's a sodium sparing mineral, cortical aldosterone. It promotes sodium absorption in the kidney and in the GI tract later, we'll see as well. So, but for now, in the salivary gland, it's not that important. But this system, you'll see again. Okay, so none of this is unique to a salivary gland. This is just the way to introduce you to it. This is the way it works. All right, so now let's go to 3. This is called an anion exchanger. All right, they didn't have a name for the other one, which is all they call it is a sodium hydrogen exchanger, right? But this one, most textbooks will say AE or anion exchanger. What are you doing again? Before you were moving sodium, exchanging sodium for hydrogen. Now you're exchanging chloride for bicarb. Okay. All right. So that's good. So you're secreting bicarb to the lumen here. Chloride goes out here and then out here. So to secrete bicarb, you're gonna need to move bicarb in. Okay. And that's usually done again. This is a co-transport system. So you're going to be doing anion plus cation. You're not exchanging, you're moving, and you want to stay electroneutral. Hopefully this approach helps you sort of, because you're going to see these again and again. And eventually it shouldn't be memorization. You're going to know chloride exchange bicarb. So you're just going to know it. You don't have to memorize. Now, how does Cftr work? It fuels. What it means by that is once this channel is open, chloride moves into the lumen and drives this particular exchanger. Without the chloride in here, you can't exchange. Okay, so that's where Cftr becomes important in fluid reabsorption bicarb. Okay, that's not too bad. Now, the only thing that they show you here is low water permeability. You have a tight junction, and usually we had water like moving across, sodium even, but here, very low permeability. They're almost, the tight junctions are so complex, they don't let water move across. Okay, there's no aquaporins here, nothing, so it's very difficult for water, right? So saliva usually is going to be hypotonic, and that's what we get, okay? Usually you're going to be absorbing more sodium chloride than you do secrete potassium and bicarb. Okay, that's written down here, good, and typed up. Now, why is saliva hypotonic? Everywhere people want to know, why is this thing hypotonic? Well, saliva is the first place where food enters or you ingest food. So it's really important for taste. And I'll explain that a little bit later. And some people think it's very important for sodium or salt detection. Animals seek out salt. Okay. You need sodium chloride. If you don't get it, you're in trouble, right? And also, you don't want to drink seawater. Sodium chloride is going to be very hypertonic. You're going to be able to taste it. But if it wasn't hypertonic, if it was the same isotonicity as plasma, have you ever tasted blood, like cut yourself? It's salty, right? Well, if your saliva was salty, you'd never taste it because you wouldn't know the difference. Does that make sense? Same thing with other tastes, something noxious. The more hypertonic it is, the better you have for taste. It's chemical, it's chemosensing, right? These cells are going to sense chemical, bitter, acidity. You ever have lemon or something? Yeah, it drives salivary flow, okay? All right, chemosensing. We'll cover that a little bit more. Is that okay for now? We're not too bad. And there's notes, try it. Now, here comes the tricky part. Now, this is how they found it out, and they want you to know this, I guess. And the same thing, by the way, you're going to get in the pancreas. So you don't lose by understanding how this works. And it deals with the rate of fluid secretion. So you see the osmolality, I wish that thing wouldn't do that. The osmolality is hypertonic at a low flow rate. And then as you increase flow, guess how you do that? You do that. Animals, I think they can do it with humans too, with certain stimuli, but they stimulate the parasympathetic. We'll come back to that in a minute. But anyway, you increase flow, salivary flow. And what happens here is these ionic composition of the salivary gland changes. So we're going to fool around with this a little bit. And ductal modification, as I mentioned before, sodium chloride is going to be absorbed, bicarbonate, potassium is going to be secreted. So at low flow rates, you allow this to work. In other words, that fluid moving through the lumen very slowly. So these exchangers can work. It can absorb a lot of sodium chloride, right? Secrete a lot of potassium and bicarbonate. Now, if you're moving very fast, these pumps can't keep up. You're just moving that fluid too fast, okay? And so it's okay. See, that explains sodium. At a high flow rate, you don't allow for sodium to be absorbed. Rather, at a low flow rate, plenty of sodium gets absorbed, okay? Now, let's take a look at this nice little cartoon. We're going to be looking at sodium, and all we're doing here, again, just sodium chloride absorbed, okay? And then the rest is potassium bicarbonate has to be secreted, okay? Now, let's say you put in 100 milliequivalents of sodium here, slow perfusion. Plenty, I overlaid this with red. I don't know if you could see that. Can you guys see the sodium chloride bicarbonate? Okay, well, anyway, I overlaid it with red so that you can see sodium absorption. When it's slowly flowing, plenty of sodium is being absorbed, that the concentration of sodium goes down to 20 from 100. I'm making this up, okay? You get the idea? Now, if you make it flow very fast, the transport is still operating, but the fluid moving so fast, you can't pick up all the sodium, right? So the sodium stays pretty high. And that explains everything. It explains chloride, okay? Chloride is secreted. So it'll increase with ductal flow rate, okay? And I mean, chloride is absorbed, sorry, potassium is secreted, and so it will decrease. And chloride is absorbed, and it'll be the same thing as sodium. Okay. Now, you can do this, practice this if you want. You'll get it. Even I get mixed up. All right. For bicarb, it doesn't follow. In other words, bicarb is also secreted. So you would expect it to behave like potassium. So it would decrease, right? And yet, if you look at this, bicarb increases the flow rate. So no matter which textbook you're reading, they're going to tell you the same thing. The parasympathetic salivary gland, they stimulate bicarb secretion. You'll see the same thing going on in the pancreas, the liver, I think, when you're dealing with bicarb. But because you're stimulating bicarb secretion, you actually get an increase in bicarb during the fast. It's secreting it, whereas when you're slow, it'll be going up, okay? And then it comes down with secretion rate. So this should match the things over here. But why bicarb doesn't fit is simply because the stimulus used to increase salivary flow increases bicarb secretion. And that also generates saliva that's alkaline, okay? It's basic at a pH of around 8 or so not neutral, it's basic. And the reason, I guess, for that is one of the major stimulants of salivary secretion is acid. You know, some acidic solution, and you're going to stimulate bicarb secretion to try to neutralize that. Does this make sense? Okay. Anyway, that's how it works in terms of that. If you want, that's why I made two figures. I don't know if I have two figures posted or not, but I'll repost this. But just play with it. You'll see that for sodium and chloride, it works. In other words, sodium goes down at low, right? At low perfusion rate, and chloride goes down. Potassium goes up at low perfusion rate. It's bicarb, it doesn't make sense. Okay. And the only reason for that is that to get that fast perfusion, they have to stimulate bicarb secretion. And anion moving like that drag water also. Okay. This is a summary of the neural control. You know, we're not going to be talking about endocrine control of the salivary gland. I don't think there is one, except progesterone is minor, but parasympathetics are going to be the major players, and that's shown here. Acetylcholine coming from the parasympathetic. It'll activate both the calcium, the cell signaling mechanism, but mostly calcium, and it'll drive electrolytes in water as well as the mucin, and we mentioned mucin, but there's also salivary amylase and starch digestion. I'll cover that in a minute. But norepinephrine or the sympathetics, they play a minor role in overall secretion. Okay. This compares sympathetics and parasympathetic influences on salivary secretion. Sympathetic is minor play. So the output of saliva is going to be very little. It's not going to last very long, transient. Mostly it's going to be protein rich, okay? Mostly it's going to drive this. Amylase is protein, right? All the enzymes are protein, right? Okay. All right. Parasympathetics are, well, if you denervate it, not much happens. Most of your drive, you have parasympathetic tone, it's driving. Your salivary flow right now is usually being driven by parasympathetic. So the parasympathetic, large amount of secretion, not the copious means, a large amount, and it'll last longer, and it's more electrolyte rich, protein poor, if you will, because it's fluid moving. And here you decrease secretion if you section the nerve, parasympathetic to the salivary gland, and you get glandular atrophy. I haven't addressed it very much, but anytime you stimulate organ systems, I'm sure got this probably in a foundation or something. If you exercise skeletal muscle, guess what happens to skeletal muscle? It increases, right? You don't exercise that skeletal muscle, it atrophies. You break a leg and you're in a cast or something, you can't move your leg, the muscle will atrophy. Okay. So it's the same thing with gland. You stimulate gland, they'll stay tone, if you will, like muscle tone. And if you take away the stimulus, they atrophy. And the same thing will be happening in the stomach. There's some pathology I deal with in the stomach, if I remember. Okay, this shows salivary flow as a function of parasympathetic activation, just to show you the range of dealing with it. Ordinarily, percent of maximum, so 10, less than 10, 5% of maximum, what you're probably experiencing right now, unless I frighten you so much with the transporter that you have fear or anxiety, you may have a dry mouth. Okay, we'll cover that in a minute. But usually resting, you have salivary flow. Okay, other moisture in your mouth, we'll come to that. Feeding, dramatic increase in salivary flow when you eat. Okay, the help chewing, we'll cover that. And noxious stimuli, right, the most potent stimulant of salivary secretion, called water rash. I think I have it written in the notes, but I don't know if you ever had something like, let's say you're almost vomiting or something, the person is going to ask you, your whole mouth's going to fill with saliva. Water rash. Okay. If we have time, we'll go back over some of these things. This is the function of saliva between meal, during meal. They have your protective and digestive. Remember, your GI tract is pretty smart, take care of things. Okay. Okay, between meals, just like right now, you need clearance. Be it food particles, leftover bacteria that accumulate. You know that when you eat, you're not eating anything sterile, right? Bacteria is coming in, right? So it keeps washing, all right? Clears the oral cavity. There's growth factors in saliva. We as humans have soap and water, but animals lick their wounds. So they have growth factors in saliva. Okay? We don't do that because we have soap and water. And also it helps with prevent demineralization, loss of tooth, or caries, dental caries, you hear that term, the cavity. Okay, it has antimicrobial action, bacteriostatic. You know what static means? It's vital. Bacteriocidal kill, bacteriostatic, it prevents them from multiplying. Static, they stay. Okay, so that's important, and you can wash them away. It's always a fight of getting rid of the bacteria versus how fast they multiply. And it facilitates speech. If you have a dry mouth, very difficult to talk. As a matter of fact, actors, when they're on stage, project and talk, speak clearly. I mumble, but speak clearly. They put pebbles in their mouth to stimulate salivary flow so they can speak more clearly. How do you do that? During meals, it's going to help processing the food, covering it, lubrication. The mucus will cover it a lot. You're going to basically eat until you're comfortable with swallowing the material. And you don't necessarily swallow all the material in your mouth. You just swallow that part that's readily swallowable. And you keep chewing on the rest. Okay. It buffers acid. We said it's alkaline, right? And also the acid, you know, stomach makes acid, right? And, you know, you can reflux from the stomach. So salivary goes down to the esophagus and helps neutralize that. Enhances taste. That's chemoreceptors, I mentioned that, taste receptors. And it solubilizes the taste receptors, and it's hypertonic, allows you to taste these things, okay? And a good example, a better example than taste is chemosensing. And basically, let's say you ingest something that tastes foul, noxious, terrible. What do? You spit it out, right? First line of defense. You know it's that. So before you swallow it, let it go to the GI. They tell you just to spit it out. Don't even bother swallowing it. Okay. If it doesn't taste appropriate to you, you're going to spit it out. It's a defense. And you notice I have it. We have growth factors, either defense, lubrication, defense. There is some digestive function, but they're very minor. What else? Can we cover? Yeah, some people think it's very important for salt protection. This sounds cruel, but in the West, it used to be people would go hunting, deer in the forest. I think they still do in Europe somewhere. But you know, they go hunting, and the way they would trap deer, they go hunting in the winter. Plenty of snow, which is, well, you don't have snow here. But what they would do is they try to lure the deer with big blocks of salt. The deer would come to get their salt. And then they shoot them a bow and arrow. I know it sounds cool, but good example. All right. Now, saliva here, you can't see it. At the top there. Doesn't go away, huh? Okay. That would be some kind of noxious stimuli. Let's see if I can. Nausea, okay, noxious, anything that, well, noxious means nauseating, right? Anything that's nauseous will turn on the parasympathetic. Here we, you know, there's not that much input from the sympathetic. They do something, okay, and both nervous systems act on the acinine ducts. And, but it's the parasympathetic system. This is from Castano. Okay. And if you're dehydrated, I mentioned that with the thirst, your salivary flow is going to go down because you just don't want to lose water anymore. Usually you don't get that dehydrated, but if you were, Saudi Arabia, it's going to happen if you're outside too much. So your salivary flow will stop once you get dehydrated. You'll have a dry mouth and you'll be thirsty. All right, makes sense? Salivary secretion also goes down during sleep, which is fortunate, and also fear, like going through this lecture. Anyway, the salivary flow will go down. Now, this is the salivation associated with feeding. You have anticipation. Now, we're going to get these phases again. They're good for talking about it, but they're continuous phase. The cephalic phase means brain, okay, going to be central nervous system, okay. Then the other phases, there's the oral phase, and other secretions will have gastric phases, intestinal phases, etc. We'll get to that later, but first I'm introducing you to the cephalic phase. Sight and smell has nothing to do with something going in your mouth, right? It's just sight and smell. You smell the food, your mouth starts to water, right? Okay, you see something that's pretty appetizing, you can stimulate the saliva to flow if it's nice, okay? Appetizing, good. And this is usually transient, and then it's reinforced by the fact that you actually ingest something. Now your taste and texture becomes important. What does it taste like? If it tastes really good, then salivary secretion keeps on going, right? But if you don't eat, it'll eventually go away, right? You're not going to salivate all day long because you thought of a good meal. Right? And then they have something called conditioning. Have you heard of Pavlov's dog? Have you heard of Pavlov's dog? Yeah. This is Pavlov, he was a Russian scientist, and it's called conditioning. What Pavlov did was presented food to the dog. He had the dog instrumented so he could measure salivary flow, okay? And so if he presented food to the dog started salivating. Okay, that makes sense, all right? That's sight and smell, and there's food there, right? But then he started, he'd ring a bell, and the dog wasn't paying attention to him. No salivation. So then he started ringing the bell and presenting the food simultaneously. Of course, the dog started salivating. And he did this, and after a while, he could just ring the bell. No food, no smell, no nothing. And the dog would start salivating. And the same thing happens to children. You may be out playing or something like that, and if your parents call you in to eat, you know food's coming. You already start salivating in preparation. Also, you have gastric acid secretion. Anyway, that's what Pavlov's contribution is called conditioning. In other words, this stimulus bell has nothing to do with the sight, smell of food, or anything that you can condition and respond to associate. And actually, CNN had an expose on this. It turns out it's not quite the same as we thought. But now, do we have time? How many? Want to go back over to transporter? Let's do that. If you already are feeling comfortable, well, don't try to leave because it'll make too much noise. Just try to sit through the acinar secretion. There's no acinar secretion. Major driver always, no matter where you are, every cell is going to have the sodium potassium ATPase, right? Yeah. All right, primary transporter. And secondary will be all of these other ones. Secondary is anything dependent on the sodium gradient created by this pump. They call it a pump. You've heard that before, right? Sodium ATPase or sodium potassium pump. It's actually an exchange mechanism, not equal. Anyway, this will allow this to operate, which is your sodium potassium to chloride transporter. Okay, this is transporting chloride. Why? You need the chloride because this thing is chloride driven. Salivary secretion is chloride driven. You secrete chloride, everything else falls into place. Okay, and that's how you know that. And secretion in the intestine will be chloride driven as well. So you'll see this often. Every time you need chloride, you want to make hydrochloric acid in the stomach, guess what you're going to need? You're going to need this transport to move the chloride. You know it makes hydrochloric acid, right? The stomach. No, you'll find out eventually. As long as I'm not over time. All right. Okay, now you move chloride in, sodium just cycles through here. It's moving this transporter. Now the chloride coming in, there's a channel for it. It's not Cftr, probably is, but it's not known for sure. It moves chloride primarily, but can also move bicarb. I'll digress here, but go straight to here. If you're moving bicarb, guess what you've got left over? Proton, right? That's going to you keep doing that, it's going to acidify the cell. So when you have this carbonic anhydrase system working, you'll see again and again, you'll see it in the stomach, in the parietal cell, same concept. Okay, everywhere you see that. If you're moving bicarb out, you're going to have to get rid of the proton that's accumulating. If you're moving the proton out, you're going to have to get rid of the bicarb that's accumulating, right? Because you move this out, you're going to have more of this, right? And it drives this. Okay. What's left here? Potassium, you've got channel. You put a nice channel in here for you. There's another one. Don't worry about details like that, but know the concept here. Sodium, hydrogen exchangers used to move it, and I mentioned. Now you're exchanging a cation for a cation. And most of the time, you're going to see hydrogen sodium exchanges working. You're going to see bicarb chloride exchanges working. That's in the next slide. All right. At any rate, you also, if you're moving chloride, let's say I turn this around, and now we're decided to move sodium through some organs in the GI, which we will. Okay, you're going to be moving sodium. Chloride is going to be moving through the tight junction, the paracellular pathway, creating a positive gradient for it to move across between the cells, not through the cells, but between. Water will move between. But here they conveniently put in an aquaporin, so there's no water. You'll know it. If someone says aquaporin somewhere, you know what it does, right? It moves the channel for water. Is there a problem with this? And it's all written out. And the duct system's the same. It's all one picture. You can do whatever you want with these PowerPoint. In the duct system, the only reason I put this in dashed line in black, because you're going to see the system again and again. It's sodium absorbing, potassium secreting kind of thing. Here it's absorbing sodium. Again, if you want, you can put that little squiggly mark in there. This is packed sodium ATPase. Every time you see it, it's going to be sodium ATPase. And then potassium channel. All right. Chloride channel, one of the major chloride channels in the GI tract and elsewhere, lung, wherever else. It's called the cystic fibrosis. If you have, if this channel is inoperative or defective in terms of operation. And you have here you have an anion exchanger, again, chloride versus bicarb. Same anion, okay? Chloride, not the same anion, but an anion for an anion, okay? Every time you're going to exchange something, usually, in general, usually it's going to be the same charge. Anyway, the big, I guess, the take home for salivary fluid is going to be hypertonic, and that allows for taste. We discussed that. And the reason it's hypertonic is because these tight junctions in the ducts are not very permeable to water. They don't have aquaporin on top of it here, so it doesn't allow you to move water. And you absorb more sodium chloride than you secrete potassium bicarb. And then this was a tricky one. You notice that even at the high flow rate, it's still hypertonic, right? You're still going to be able to detect taste, okay? It's just going to be high flow rate. The osmolality is trying, you know what normal, it was about 280, what isotonicity of plasma, usually around 300, anywhere between 280, 320, that's what most textbooks will use. But it's still hypertonic. And then this was a little tricky. Want to go over it again? The duct flow, or do you know it? Does it make sense to you? slow perfusion. If you're absorbing sodium and chloride, then the same thing's going to happen to sodium and chloride. So sodium come in if it's very slow. You haven't activated your purpose right now. Let's say slow perfusion. You're going to have, well, it's going to be blood-like or plasma-like. It comes in, and then you're going to absorb almost all of your sodium sparing, if you will, and it's going to be very hypotonic, but also less sodium. And if you have fast flow, then you don't have enough time to reabsorb the sodium, and sodium is higher. Does that make sense? And so that describes this flow pattern. That's how they came up with all of this, by doing these kind of estimates, micropuncture. Okay. You know, a micropuncture, you take a little micro needle or whatever, they can puncture the duct. Okay, chloride is absorbed, so it's going to behave the same way that sodium is. Okay, if you increase perfusion, you're going to have more chloride at the end. Now, the secreted is going to be just the opposite. You increase the flow rate, you're secreting, you're adding. You don't have enough time to add it. So it will decrease with increase. Does this make sense? Anything that's being absorbed will decrease with increased flow. And then I showed you bicarb. It's a prediction for bicarb because that's what, if it was just a transporter operating, then you would expect it to go down, go up, stay up here, and then go down here, but it doesn't. And that's because the major mechanism of increasing flow in the salivary gland is through the parasympathetic, and they stimulate bicarb secretion. So that's why that. Is that okay? Okay, I'll see you tomorrow.